Alright guys, welcome to video 5 of unit 3. We are putting together these past two videos about transcription and translation and putting it all together to talk about the Central Dogma as a whole. So, just to recap kind of what we've been talking about. DNA is a double helix string of base pairs that codes instructions for building proteins. So when we say base pairs, we're talking about that A goes with A, T, C, G. Those are the base pairs. So you guys know DNA is like a long string of all these base pairs, and they code instructions for building proteins. We're going to talk all about that process in this video. That process includes these two steps: transcription and translation. Transcription being where DNA creates a copy of the instructions into mRNA, and then that mRNA travels outside the nucleus to the ribosome and forms a protein. So before we get into it, just to recap, make sure we're on the same page here. Why is mRNA important? So yeah, DNA is inside the nucleus here. It cannot leave the nucleus. DNA cannot leave the nucleus. So it has to create a copy of its instructions in the form of mRNA, and then mRNA is able to leave the nucleus and go to the ribosome. Oh, that's horrible handwriting. mRNA goes to the ribosome, and that is where the ribosome can translate the mRNA into amino acids and forming a chain of amino acids that gives us a protein. We're going to get a little more into that now. So let's say this is our DNA strand, and you guys know these base pairs. A goes with T, C goes with G. So on our DNA, if we have a C over here, what would be on this side? Yes, that would be a G that fits together with the C. That's what we mean base pairs. They pair up like this. So if this was our first DNA codon, our mRNA would find the matching pieces and start building that mRNA strand. We're going to look at these three codons in the DNA, and what would our first mRNA piece look like if this was our first codon right here? So obviously a C transcribes to a G transcribes to a C, and then an A in mRNA transcribes to a U. You guys will remember that all the bases are the same, A, C, G, except for no T. mRNA does not have T as one of its bases. So when we're transcribing DNA, we have to remember that T is not an option. So what would the next codon here transcribe to? So this would be our full mRNA transcription piece based on our DNA. So then this mRNA is able to leave the nucleus after it has these instructions, and it travels to the ribosome. So the ribosome's job is to translate each codon or each set of 3 mRNA base pairs into an amino acid. Each set of 3 translates into one amino acid. So if we're looking at our first codon here, GCU, we're going to take a look at the codon chart, and we'll see first letter G, second letter C, third letter U right here, GCU. On our codon chart, that's shortened to ALA. That stands for alanine, but you guys don't really need to know what the names of each amino acid are. You can usually just go by the 3-letter abbreviation. So our first amino acid here would be alanine. Then we go to our second codon, UCG, and we're going to see what amino acid that translates to. So looking here, I see UCG translates to SER. So that would go ahead and get added to our amino acid chain. Finally, we have AUA. That translates to ILE. So then that would become our final amino acid on our chain. Proteins are actually much longer than only 3 amino acids, but just for this example, we're looking at as if this were only 3 amino acids long. Okay, we're going to look at a brief example of a specific protein. So this is the MC1R protein, and it allows the body to produce eumelanin, which gives color to the skin and hair. So the real MC1R gene has 954 base pairs, but we're obviously not going to look at 954 base pairs in this video, so we're going to just look at a small section of it. So looking here, we're obviously in the nucleus because we're looking at DNA. We see these 3 codons that are here in the nucleus. Our first codon is CGG, which transcribes to GCC in mRNA form. If you keep going down our DNA to our next 2 codons, you can see what they transcribe to here for mRNA. Once the mRNA is formed in the nucleus, it travels to the organelle that builds proteins. That organelle is, of course, the ribosome, and the ribosome kind of wraps around the mRNA like this and goes codon by codon. So 3 bases at a time, the ribosome reads the mRNA and translates each codon into an individual amino acid, forming a long chain of amino acids. So our first codon here is UGU. So we're going to look at our codon chart, and find that UGU codes for CYS amino acid, cysteine. So the CYS amino acid is brought to the ribosome by something called a tRNA, which is a transfer RNA. Next up, the ribosome is going to move along to the next codon. This codon, AAC, codes for amino acid ASN, so that amino acid gets added onto the chain. Our ribosome is going to move right along to the next codon, GCC. So looking at our amino acid chart, what will our amino acid chain say once the ribosome has translated the codon GCC? GCC codes for amino acid alanine, ALA, so that gets added on to the end of our amino acid chain. Like I said before, the real MC1R protein is coded for by 954 bases in real DNA. So obviously this would continue for a long, long time until we had a protein created that looks like this. The MC1R protein that creates the trait of darker hair. So in reality, there's not just one version of this gene. That would mean that everyone had the same exact color hair. There are over 200 known versions of the MC1R gene. So that means that there are over 200 different levels of this protein production you could have. There are 200 different ways that your DNA could code for different amounts of this protein to be produced. So if you have a gene for MC1R that causes less protein production, then that means that you would have lighter hair. But if you have a copy of the MC1R gene that causes a lot of this protein to be produced, you would have darker hair. These different versions of the same gene are called alleles, and that's something that we're going to talk about a lot more in the future. Right now, we're just focused on how DNA goes from just a string of letters in the nucleus to building proteins that cause traits. So key points here. Sections of DNA are transcribed into mRNA. mRNA then travels to the ribosome and it is translated into amino acids, which form proteins. These proteins cause the organism to have the traits it does, including diseases, appearances, everything. This visual is the one that we're going to show the most in class. So taking a look at this can be super helpful. Also, if this visual isn't the most helpful for you, looking up the Central Dogma visuals, diagrams, etc. There are a lot of resources online about this, so I'm sure you can find something that will be most helpful to you.